Hello everyone, I'm called Okumu Geoffrey, a member of Group A today. We are trying to, I'm trying to review a paper called Explainable Hybrid Vision Transformers and Convolutional Network for Multimodal Glioma Segmentation in Brain Magnetic Resonance Imaging. Brain magnetic resonance imaging is a machine, or is the other, and it's a machine used for actually a diagnosis of all generally for general scan of the body, and including of course the brain. Actually it is used for major scans. The explainable hybrid vision transformers, here we are trying to look at the explainability of the model that we are going to use, since in the medical sector there is high need to give results which are backed up by the explanations of how you reach out to the result. That's the reason as to why they are trying to build an explainable model. And of course, our topic is called machine vision for identifying medical plant species. It's a topic, it's a project that we are trying to work on, and we are going to use image of leaves in order to detect the different plant species or the different medical plant species within the globe. And the computation problem here is accurately segmenting gliomas, which is a type of brain tumors within the brain. Brain tumors, I can refer them to tissues within the brains, like abnormal tissues within the brain, which can become either cancerous, or some may be non-cancerous, and of course others can be treated as well. There is also, other than detecting the brain tumors, we are also trying to look at the explainability of the model or the logic behind the result that is produced. The proposed machine learning models are convolutional neural networks and the vision transformers. We shall look at how they work. And of course, here we are trying to use the vision transformers in order to, before we use vision transformers, of course, the image is passed into the convolutional neural networks layers, the different layers, that is from the input layers, to extract the different local dependencies like color, the tumor age, and many other things that are within the scope of all the regions within the brains which are related to tumors. Uh, it feeds these extracted dependencies into the vision transformers, which later on uh, breaks them down into patches and rearrange them in order, actually breaks them into patches, analyze the relationship between them, and rearrange them in order for them to be evaluated. The segmentation result, although it doesn't look so clear, here we have taken samples, two different samples from, rather three different samples uh, from the two categories, that is the low grade glioma and the high grade glioma, which is on the right, which for high grade glioma. This, as we are all aware that MRI, which is the magnetic resonance imaging, takes their images into all the scans is done, but it gives the images in different modalities. That is the FLAIR, which is the fluid attenuated uh, inversion inversion recovery, and there is T1 weighted, there is T1G D weighted, there is T2, which is this line. There is the This is our prediction, which is for the trans XAI, and these are the truth or the ground truth. So basically, if you look at them very well, you won't see a much difference in the result that has been got out of it. The evaluation metrics used, there is dice similarity coefficients, which measures the overlaps between two different sets of uh, of data. For example, for this case, the sets of data that we have, they are the ground truth, and of course the prediction that we have got out of the models that has been built, that is a trans XAI. Uh, and of course you compare and see the results. We shall look at some of the samples of the results that we have got. There is also Hausdorff distance, which deals with the maximum distance between the two sets of data. And of course, here also we are trying to talk about the ground truth and the segmented results, which have been produced by the TransX AI. This table one shows the sample results that has been got out of this the experiment that has been done, uh, and of course shows they use a five-fold cross validation step. This five cross validation is used in a scenario where there is limited, or the size of the data set is small, and yet you need a, a relatively large size of the data set. So you do things instead of doing augmentation, rather data augmentation and all those other stuff. You can also try to break down these data sets into different folds, which for this case has been done into five folds. And you can use these data sets interchangeably in such a way that you can use some for training, others for validation, as well as even testing. You be rotating them like that until all the five folds have been used for training and validation, in order to see how how they are, or how the variations between the results that has been got. Here they did with the HD95. When you see low, for example, 4.3, this one 7.7, like the low HD95 figure shows that there is a good result, or the result got is more or less closely to the expected outcome of what we are supposed to. Meanwhile, whereas this one shows in terms of probability, where the higher the figure, the higher the rate of, or the higher the the similarity between the result. This comparison was done with different other methods, or the different other architectures. So you can pass through them. You can see there is this one which has this comparison was done with other institutions, which some of these institutions were part who used the, like for example, institution one used the same data set, which is the brain tumor segmentation 2019 overlap. The saliency map here, we are trying to look at the visualization got from the different from the TransX AI, in such a way that it is trying to give us the result. These results are based on these results are in white and gray matter. There is a flare MRI, initial tumor localization, the all those aspects which have been seen in there. And of course, the implicit concepts here means that the model actually went ahead trying to explore more and give out a visualization beyond what has been trained, what it has been trained for. And of course, the explicit concept is referred to the ones which is the exact expectation that the model has been trained to do. So just that is the difference between the two. And the impact of this input modalities, as you can see here, you can see the variations in the different you can see the variation in the different samples that you can see, the pictures you can see there, the edema, the enhancing tumor, the necrotic, the MRI, and the truth, of course, are the line which is on top here. And also this line is for the truth value. As you can see, this one is for T2, T1GD, and of course the other ones. We are trying, these are the real architecture of how this model works. An input is fed, which is an MRI data, which is an image. And these images, we can see that this CNN has around eight blocks, or eight layers, which these eight layers helps in the extraction of the local features from the image. And this feature is passed through this part, which is the vision transformer. And of course, it tries to extract some other images. Of course, it breaks down the images, the features into patches, and of course, relate it to what we have here in the we have what has been what is expected out of it, and of course, this is the visual expectations that we are in need of, and this architecture is also compared with some of the experts knowledge medical knowledge in line with the what we are trying to do. This other architecture goes deeper and of course breaks down when this image is fed into the into the system. The system takes it as an input. The model, which is the CNN, the upper layer, of course, use this uh, downgrades it, and of course extracts the necessary features that it is supposed to extract, that is the local features like the color, the tumor size, the age, and everything, uh, and also brings it into this vision transformers block. From here, it is broken down into patches, and of course these patches uh, are having also layers, which we can see here ln, which is the layer normalization that helps in normalizing the activation across each of these features. uh, After this normalization, it is passed into the multi-head attention. From here, um, we try to see that uh, in the core components of the vision transformer architecture, it allows models to attend to relevant information and consider the relationship between the patches within, and captures the global dependencies, of course, from here, and understand. Here it is tries how does it understand it? It tries to relate between the different patches that it has extracted from the previous features from the CNN layer. Then from here, which is this part called the multi-layer perception, they feed forward neural networks that further processes the information. Is now from here, the patches, the different patches that have been extracted, they are related to each other in order to discover the nonlinear relationship between them. And of course, all these are combined back and taken back to these remaining four blocks of the or the four layers of the CNN. And here we are now trying to work on the explainability using the Gradical. Uh, I didn't talk about it previously. Here we are trying to employ the post hoc explainable AI method that that is uh, that is used in order to explain this model. All along, the image, the input image is passed through this uh, this forward pass, and of course from there, when it is passed there, the segmented patches or the different here the input is passed through the forward pass, and here of course it predicts and gives the segmentation result from here. And this segmentation result is compared with what was supposed to actually come out. If there is some difference or there is some disparities, it is propagated back to the model in such that the biases is also shown, and also the what is expected is shown and is compared. This is done by the Gradicam, and of course, it gives back it helps in improvement of this model for it to in order to learn. The data set that has been used, I've been talking about some of these things. This is the data set. It is called Brain Tumor Segmentation Challenge 2019. It is a data set uh, that is containing images or MRI images from different institutions, around 19 different institutions. The strength is explainability. Since we have looked at this, we have realized that it is able to explain. This model is able to explain beyond helping the experts, and even making the experts to understand, and of course help them to trust this trans XAI. Of course, as I talked of earlier on, they use post hoc explainable artificial intelligence. I would talk much about it, but because of time, I would advise you go and read much about it. The weakness is the explainability is just within. It's only there is only visual explanations, which does not actually, of course, helps and give all the details of everything. It depends on the interpretability of the person reading your visual explanations or the heat maps. The current implementation is only on 3D and limited to 3. Cannot process the 3D images. This is the reference to these images, rather to the different resources I used in order to come out with this presentation. Thank you very much. For question, we will post in the chat section.